Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next exam. Still on the business of Waec 2024. This time around, talking about biology practical, the fast approaching biology practical. Having looked at specimen A to E, when we look at the list of specimens, specimen F, G, H, J, and K falls into another category of specimens. This time around, we are looking at storage organs. Yes, we're looking at storage organs in plants. We're looking at sort of a natural vegetative propagation and the likes. Okay, let's get right into it. Specimen F is ginger rhizome. Specimen G is onion bulb. Specimen H, leaf of a bryophyllum, and all of that. Okay, the first question that pops on your screen talks about identify specimen F, G, H, J, K. But in identifying specimen F, G, H, K, you may have the question coming as identify the type of storage organs of F, G, H, J, K. Specimen A is a rhizome. Specimen G is a bulb. Specimen H is a bryophyllum. Specimen J is a stem. And specimen K is a tuber. That's the type of storage organ that specimen F, G, H, J, and K are. And then the next question says, name the part of the various plants that we have listed in this specimen in which food is stored. Name the part of the plant in which food is stored. So, specimen F is a rhizome, is a storage organ called rhizome. G is a storage organ called bulb. H is a bryophyllum leaf. J is a cassava stem. K is a cassava tuber. So, what are the storage organs of these various specimens? F, which is a rhizome, food is being stored in the stem. For the bulb, talking about the onion bulb here, food is being stored in the leaves. Yes, food is being stored in the leaves. You see it on your screen, the fleshy leaves. Food is being stored in the fleshy leaves. Okay, talking about bryophyllum, food is stored in the stem. When you talk about the cassava stem, it is actually stored on the roots. And when you talk about cassava tuber, the food is actually stored on the stem. So that is how that works. Another question coming up is that we should name 3 parts common to structures F and G. 3 parts common to structures F and G. Okay, talking about structures, specimen F and G, the 3 parts common to them, number 1, adventitious root. As you can see in the diagram on your screen side by side, they both have adventitious roots. Number 2 is that they both have modified stem. In the case of the onion bulb, you're going to see the compressed knob-like stem. And for the rhizome, you're going to see the rhizome, that's the modified stem. So they both have stem, and also they both have leaf. You can see the green leaf on for all of them, both the photosynthetic leaf and the fleshy leaves. You can see that. So they both have leaves. That is something common to specimen F and G. At least 3 parts common to structures F and G. That's to specimen F and G. Then to the next question, question 4, it says, suggest one method of propagation of F, G, J, and H. F, G, J, and H. F, G, J, and H. We should suggest one method of propagation. Yes, for F, when F is going to propagate, when it is going to sort of reproduce, it is going to be via the rhizome itself. When it's specimen G, it is going to be via the bulb. When it's specimen H, it is going to be via the leaf. Yes, specimen J is going to be via the leaf. That's talking about the bryophyllum. You see on the bryophyllum, you're going to see tiny bryophyllum arising from the leaf itself. Yes, with adventitious root all budding off from the leaf itself. That is why the method of propagation here, look at it, they said we should suggest one method of propagation. It is via the leaf here, and then the last one talking about cassava tuber, it is via stem cutting. That is the method of propagation for this. So you're looking at, if we are going to propagate specimen F, is going to be via the rhizome. If G is going to, propagate itself, it's going to be via the bulb. If H is going to propagate itself, if H is going to propagate itself, it's going to be via the leaf. And if K, cassava tuber, is going to propagate itself, it's going to be via stem cuttings. And that ends question 4. Can we be told to draw anything from this specimen F, G, H, J, and K? I don't think so. It's a no from me. I don't think that's going to hold. Okay, moving on to the last question. The question says, as you can see on your screen, examine specimen F, G, and J and comment on their features of biological interest. That we should comment on some of their features that are of biological interest. Okay, starting with specimen F, as you can see in the diagram on your screen, it is the rhizome. It is a rhizome. As you can see, the various labeled parts, you have the terminal bud growing upwards, you have the lateral bud growing sideways, the various parts, the fleshy stem, the nodes, and the adventitious root, and all of that. So what is of biological interest here? Okay, you can see on the board, specimen F is a thick, fleshy underground stem which grows more or less horizontally beneath the surface of the soil. Some of the food manufactured is usually sent to the lateral bud. It's usually sent to the lateral bud. That's very important. It's usually sent to the lateral bud. You can see the lateral bud in the diagram there. It's usually sent to the lateral bud, which continues the growth of the rhizome. It is the lateral bud that will continue the growth of the rhizome. When the older parts of the rhizome die, it is the lateral buds that will grow into a new aerial shoot. So it is like this. Out of the food manufacture, some of them are sent to the lateral buds. The remaining ones stored. When the older parts of the rhizome die, it is the lateral bud that will grow to form a new rhizome. That is what is of biological interest. That's a feature that is of biological interest. That when the rhizome dies, it is the lateral bud that is going to develop into a new aerial shoot. Hence, food is usually being sent to the lateral bud. So that ends specimen F. So talking about the biological interest of specimen G, that's the onion bulb. As you can see in the diagram on your screen, you can see the compressed stem, you can see the terminal bud, and you can see the lateral bud. It is the lateral bud that breaks away to form an independent bulb. So that's the most important thing we need to understand here. Talking about specimen G, onion bulb, they have swollen on the ground, compressed or knob-like shoots or buds with inner scale leaves, which are fleshy and swollen with food reserves. You can see the fleshy leaves, they are all swollen with food reserves. Now, a parent bulb produces one or more bulb which breaks away. Now, the leaves of the terminal bud, you can see the leaves of the terminal bud grow up to manufacture food for the formation of a new lateral bud. That's by the side, lateral bud by the side. It is this lateral bulb that now breaks away to become an independent bulb. So that is the biological interest of specimen B, is the fact that the lateral bulb breaks away. Lateral bulb which breaks away to form an independent bulb. So to manufacture food for the formation of a new lateral bulb, it is this lateral bulb that now breaks away. That's why you see your onion, when it's going to be, you see some of them, they look as though two. They are always breaking off by the side. So what we have here is that the lateral bulb, which breaks away to form an independent bulb. So that ends specimen G. Okay, talking about specimen J, we are looking at cassava tuber, and if you look at the tuber, usually on the ground, it has a terminal bud and then the axillary bud, otherwise known as the eye of the tuber. Yes, the eye of the tuber. Now, when you talk about specimen J, it is a woody surface stem with buds or nodes, which obtains nutrient from stored food in the terminal end of their underground stem. Okay, there is the terminal end of the underground stem, and that is where they obtain food from. So that is just about simple and direct, and that just ends that talking about the biological interest of specimen F, G, and J, and that just about ends that question, talking about features of biological interest for specimen F, G, and J. And of course, aside specimen F to K, we still have a specimen L, M, P, Q to go, talking about the thoracic vertebra, humerus bone, femur bone, lumbar vertebra. Are we having any drawings here? No, I do not predict any drawings from here, but in the next set of specimen, about 4, talking about the thoracic vertebra, the humerus bone, the femur in the thigh, and the lumbar vertebra, we are going to be having drawings there. And of course, not to forget the last 2, mango seeds and orange seeds. Do not forget, hit the subscribe button, share with your peers. From myself and the entire team at 300 Plus Academy, it is bye for now.